Good morning, good afternoon, good evening, everyone. My name is Mike Hodgson. I'm a senior manager at Baxter and lead the global E&L community of practice. Thanks for the opportunity to talk to you all today about some of the technical challenges associated with large volume parenteral and complex drug products. I think this symposium is long overdue. I think there's a lot we've got to talk about, and I'm going to try today to give you a high-level overview of some of the challenges that I feel as an industry we need to start talking about and find solutions for. I've been very fortunate in my career to date in the terms of having worked for three fantastic companies that all focus on different product types, and really that's given me a unique perspective in terms of extractables and leachables, and how you might approach each different product type in a slightly different way to address the risk and put the patient at the center of any strategy. So I hope to share that perspective with you today and drive a discussion in the future. Okay, so this is a picture that I feel sums up E&L. And I feel quite passionate about this in that really there is no one size fits all approach to E&L. Different products have different challenges. And it's important that when you are developing your E&L strategy for a given product, you think carefully about what the risks are, and then you address those risks specifically, rather than applying a very generic approach, which might be appropriate for one given product type, but may well miss the key risks and address the key risks for another product type. So some examples here, 3 different product types: metered dose inhalers, topicals, ointments, creams, and large volume parenteral drug products. So metered dose inhalers are widely regarded as the product that presents the greatest risk from a leachables perspective, and historically have been recognized as the most challenging. That's primarily because of the formulation of the drug product being highly organic in nature, and that results in significant leachables in the drug product that are then dosed to the patient. So in order to control the risk to the patient for a commercialized product, there's a significant amount of effort that needs to go in from a technical perspective. Conducting exhaustive extractions to understand the total amount of each of those extractables in a material allows you to develop a control strategy to control those extractables in that material, and therefore control the leachables in the product by an extractable leachable correlation. The leachable profiles, as I said, are generally quite complex. There's a lot of leachables, and they're there at levels that are significant and need to be quantified via highly accurate methods to allow you to conduct a tox assessment on them. And then the implementation of a control strategy is challenging in itself. Topicals, ointments, and creams, the formulation here is a little bit different and often complex, often non-aqueous. So the impact that has on the extractable study design is significant. How do you try and mimic that aqueous, that non-aqueous formulation, and in a way that the solvents chosen for that extractable study are analytically amenable to some of the techniques that we use. And then the leachable strategy for topicals, ointments, and creams becomes challenging because of that formulation. There's a significant amount of effort needs to be put into the sample preparation to ensure that you can extract some of these leachables that you may well be interested in from the cream or the complex non-aqueous formulation into a solvent system that is then amenable to either gas chromatography or liquid chromatography. And then large volume parenteral drug products. I'll go through some of the challenges today, but just at a high level, some of the large volume parenteral drug products have poorly defined maximum daily doses, which creates some ambiguity in terms of how to derive an AET. Because of the very nature of a large volume parenteral, the methodology and the levels at which we are striving to detect these extractables and leachables at requires ultra trace level methods. And with that comes an increased chance of unknowns, and then having to manage that risk with a toxicologist becomes critical to providing a robust and comprehensive assessment of the risk. So really, the take-home message from this slide is each dosage form has its unique set of challenges. And really, it's not appropriate to apply the same generic approach to a variety of drug product types. And here, we'll touch on some of the challenges around large volume parenterals, but even small volume parenterals versus large volume parenterals, they present different challenges. And so it's not unreasonable to think that they should be approached in subtly different ways. So here's a high-level overview of the process of addressing the risk of leachables in a given drug product. So starting off with the container closure system and material selection, that is the point as an analytical and E&L SME which you should get involved in the process. And this is even more important for large volume parenterals. Because you're working at ultra trace levels, the cleanliness of the material is absolutely critical. The cleaner the material you can use, the increased chance you'll have of minimizing the amount of leachables in your product and therefore demonstrating safety. Having and using a stopper that has a coating applied to it, which intentionally minimizes the leachables from that stopper migrating into the drug product is also a worthwhile consideration at this stage. Anything that you can do to either minimize the total amount of extractables in the material or minimize the likelihood of them leaching into the product is worthwhile considering, as I say, because you're working at ultra trace levels. So I touched on the maximum daily dose, and we'll go into that in a little bit later on in a slide. But it's a key input into the strategy very early on. It's used to define the analytical evaluation threshold, which is the threshold we use from an analytical perspective to understand the toxicological significance of either the extractable profile of the material or container closure system or the leachable profile of the drug product. And it's really central to the strategy. So a lot of the challenges around large volume parenterals relate back to that maximum daily dose and the analytical evaluation threshold. So early on in the process, it's typical to perform an extractable study. And this is an analysis of the container closure system to establish what leachables may leach into your drug product and therefore predict the risk. And it's important at this stage to set the analytical evaluation threshold at a level that is conservative from a toxicological perspective. And in order to do that, we need to refer to a safety concern threshold, which I'll go to in a while. But what that needs to do is demonstrate that your methodology is able to achieve that analytical evaluation threshold so that you can operate at that toxicological threshold. When you've generated your extractable data with a reporting limit aligned with your analytical evaluation threshold, you then typically work closely with a toxicologist to establish the risk of those extractables that are detected above the analytical evaluation threshold, and start discussions around what the significance of any unknowns are, either that are above that threshold or below, and also start to think about how those unknowns may be addressed if analytically it is incredibly challenging to identify them. And then the output of that tox assessment is essentially an indicator of what the risk is, or should be an indicator of what the risk is from a leachable perspective if you've designed your extraction study with that in mind. And then you can use that knowledge to design your leachable study. What extractables present enough of a risk to be targeted as leachables in the product? How am I going to design my leachable study? Um, what methodology am I going to use? Am I going to align with the AET? And how am I going to manage the sample preparation with respect to the drug product formulation? So really the take home here is the analytical evaluation threshold is central and critical input when designing an E&L strategy for really any drug product type, but most importantly for a parenteral drug product, it becomes really quite critical because it often results in a very low AET because of the high maximum daily dose volumes that a large volume parenteral drug product has. Okay, so now we're going to get into a bit of the specifics around the challenges specific to a large volume parenteral drug product. So in September last year, the PQRI, Parenteral Drug Product Working Group, released an update. And they're proposing a safety concern threshold of 1.5 micrograms per day, and that was fixed. It didn't take into account whether the drug product was an acute or a chronically dosed drug. And neither did it take into account the daily dose volume, whether that was a small volume parenteral or a large volume parenteral. And I'll start by saying for small volume parenterals, that's entirely appropriate and analytically possible. But as we'll get to later in the slide, for a large volume parenteral, it becomes challenging to align with that 1.5 micrograms per day from an analytical perspective, the higher the maximum daily dose is. So as most of you probably know, the safety concern threshold is used to calculate the analytical evaluation threshold. It's derived using the maximum daily dose of a drug product and expressed in analytical meaningful units. For example, micrograms per mil, if we're talking about a unit for leachable studies, or for extractable studies, a common unit might be micrograms per stopper or micrograms per gram. So since the AET is related to the MDD of a given drug product, the higher the MDD in terms of mils per day, the lower the AET. And the lower the AET, the greater the analytical challenge. And in some cases, aligning with the AET is beyond even the latest state-of-the-art instrumentation. And we'll touch on that in the next slide. But the take home really from this slide is that the higher the MDD, the lower the AET, and the greater the analytical challenge. So before we get into the definition of the AET, I just want to provide you here with an example of a product that has a poorly defined maximum daily dose. And if you've got a poorly defined maximum daily dose, given that's an input into the analytical evaluation threshold, that creates ambiguity and challenges with respect to defining that and deriving your AET. So this is an example: sodium chloride injection. It's an IV fluid. The product labeling states the choice of product, dosage, volume, rate, and duration of administration is dependent upon the age, weight, and clinical condition of the patient and concomitant therapy. and administration should be determined by a physician experienced in intravenous fluid therapy. So there is no defined MDD. So what do we do? Well, in this situation, it's really quite critical that you involve a physician or someone with clinical expertise to advise on what is typical clinical practice. Because if you apply worst case, the maximum daily dose could be up to 10 liters, and trying to detect 1.5 micrograms in 10 liters is analytically impossible if the intent is to use a screening method. So in practice, the dose of sodium chloride is highly variable and dependent on patient needs. And we've got some examples here: hypovolemic shock, maintenance fluid therapy, and when it's used as a diluent in medications. It can result in massive volumes of sodium chloride administered in a 24-hour period. So deriving an AET for sodium chloride injection is reliant on clinical assumptions being made and expertise with respect to how that's delivered to a patient. So there are more examples, but I feel that this really highlights the challenge quite well. And really the take home here is that the MDD of some products cannot be defined, resulting in the need for clinical expertise and assumptions to be made. So when we think about large volume parenterals, we shouldn't just think about IV bags. Vials can result or can contain drug products that have a high maximum daily dose volume. And an example is paracetamol. It's available in a 100 ml vial. The maximum daily dose of paracetamol IV is 400 mils. So you're talking there of 4 times 100 ml vials that would be required to meet the MDD. So we shouldn't forget about vials when we think about large volume parenteral drug products. And when we think about vials, there are some products that are available in multiple strengths and multiple sizes, which again can bring complexity to the derivation of the AET. And here is just an example. It's not related to any given product as such, but I think it highlights the challenge quite well, in that we can have a drug product that's available in two strengths, 2 mg per mil and 5 mg per mil, and it can be available in different vial sizes: 1 mil, 5 mil, 10 mil for the 2 mg per mil strength, and 1 and 2 mil for the 5 mg per mil strength. That in itself, because of the different strength, results in different maximum daily doses in terms of volume. So for the 2 mg per mil strength, the maximum daily dose volume is 100 mil. For the 5 mg per mil strength, the maximum daily dose in mils will be 40 mil. And that then can be compared to the vial size to derive how many vials you then need to achieve that maximum daily dose. And in the case of the 2 mg per mil product that's filled into a 1 mil vial, that could in theory be 100 vials per day you would need to achieve that MDD. But in the case of the 10 mil fill vial for the 2 mg per mil strength, you would only need 10 vials. And you can see here when you're deriving an AET to assess the extractable data that you generate on a per vial or per stopper basis, that AET differs quite considerably by nearly an order of magnitude. So if you were to take the 100 vials per day example, you'd be looking at an AET of 0.02 micrograms per stopper or vial. Whereas if you took the 10 mil fill volume to derive your AET, you're talking of 0.15 micrograms per vial. So if there is reason to believe that the patient would require the MDD, should the AET be based on a situation that is worst case but clinically impractical? i.e., a patient receiving 100 vials of a 1 mg per mil product, or one that is typical clinical practice, where if you knew the patient was going to require the MDD, you would use the higher fill volume vial, or even potentially the higher strength. So again, another level of complexity that's added when deriving the AET for drug products that are available in multiple strengths and multiple sizes. So take home here really is some parenteral drug products have multiple AETs. Discussion with regulatory authorities or someone with clinical expertise is a necessary step to balance practicalities with risk and clinical benefit. Okay, so we've talked a little bit about the AET and the MDD and that and the SCT, and how all of those come together to provide you as analytical chemists with a target level of sensitivity. But I'm going to say even the latest state-of-the-art analytical instrumentation and accompanying sample prep are unable to achieve the sensitivity requirements to align with 1.5 micrograms per day for some high daily dosing LVP drug products. And I used saline as an example. In theory, up to 10 liters could be dosed in a day. Here I've based it on a 5-liter maximum daily dose LVP drug product. You're looking at sensitivity in the region of 300 parts per trillion if you were to align with 1.5 micrograms per day, which analytically is not possible if you don't know what you're looking for in your sample, which an extractable study is. You're using screening methods that are designed to be able to detect and estimate the level of a wide variety of compounds, rather than highly optimized methods that focus on a known compound that you've tuned the instrument to be able to detect. So here, this table provides an indication as to what's analytically possible with state-of-the-art instrumentation, what's possible with maybe some sample prep, and what's simply not possible from a screening method perspective. So we've got the SCT remains constant in the first column. The second column is an increase in maximum daily dose in mils, which goes from 10 mils up to 5 liters. And you can see that the AET goes from 150 parts per billion to 0.3 parts per billion, or 300 parts per trillion for a 5-liter LVP drug product. So I think those AETs that are in the green box, down to 30 parts per billion, I think are achievable with the state-of-the-art instruments that are available nowadays. Screening methods should be able to hit that level with the right sample preparation. Once you start going below 30 parts per billion, between 20 and 3, I feel it very much depends on the sample and what interference you may have from the sample and the solvent you're using, and also the sample preparation you're using. And once you start to get into the 2 parts per billion and below, really, from a screening method perspective, you cannot achieve those levels. You can only achieve those levels if you know what you're looking for, and you can highly tune the instrument to target that one individual compound. So that's an important point to note and think about, because we as an industry need to start thinking about, in those situations, how do we support the safety of our products from an E&L perspective, because analytically it's not possible to provide the data aligned with 1.5 micrograms per day. Okay, so we've talked about what the method, um, typical screening method capabilities are in terms of sensitivity. I'm going to talk a little bit now about how that varies, and how you can improve it. So I think vendor to vendor instruments really, you don't get a significant increase in sensitivity. I think there's minor differences now between the main vendors in terms of what a GCMS can achieve in terms of sensitivity. So switching from one vendor to another, I don't think buys you orders of magnitude increase in sensitivity, which is what we really need. Sample preparation, well, yes, that can. Enriching your analytes in your solvent extract, or your drug product is possible, but it's important to note that this process is somewhat selective. But there are common techniques that are available and in use from an E&L perspective. Static headspace is an example for volatiles, and liquid-liquid extraction is an example for semi-volatiles. And in particular, liquid-liquid extraction provides you with a means to enrich your analytes in your sample to achieve lower detection limits. The choice of internal standard is critical, when defining the sensitivity of your method, and it's not fixed. It varies from one organization to another, depending on the compound selected and its response factor. This can result in an overestimate or underestimation of sensitivity. If company A chooses an internal standard with a very low relative response factor, then they're going to underestimate the sensitivity of their method relative to a lot of the commonly seen extractables and leachables compounds. Company B might choose a internal standard that has a higher response factor, and this could be 100 times higher, and that would result in an overestimation of the sensitivity of their method. And if the response factor differences are circa 100, then that same method run at different organizations would result in a 100-fold difference in terms of what sensitivity they are claiming. There's various ways in which you can define the sensitivity of a screening method. There's no right or wrong way. They're all appropriate, but they result in different outcomes. So you can define the sensitivity of your extractable screening method using the internal standard, that you add to your solvent system. You can define it by the lowest estimated compound detected with a signal to noise greater than 10 using the response factor of the internal standard. So that essentially means everything that we feel is an extractable, i.e. it's got a signal to noise greater than 10, we'll integrate that peak, we'll estimate the response, so we'll estimate the amount of that using the response factor of the internal standard, and then that will produce an amount, and that amount will be an indicator of what the sensitivity of the method is. Taking the lowest amount across all your extractables gives you an idea of your sensitivity. Another way is you could derive a list of, you could come up with a list of compounds with diverse functionality. You could inject them into your system at various levels, and then extrapolate down to establish what the mean theoretical limit of detection is. And then that mean theoretical limit of detection across a range of analytes is your sensitivity. So really the take home here is that the sensitivity extractable study method varies from one compound to another. It is therefore only possible to estimate the true sensitivity of an extractable method. Okay, so thinking about the extraction study itself and the conditions that we routinely use, there's a big advantage to replicating the interaction between the container closure system and the drug product solution, as that's the indicator of what patient risk is. A large volume parenteral drug product is rarely exposed to temperatures exceeding 40 degrees C. So subjecting that container closure system or stopper to temperatures above 40 degrees C may provide you with extractable data that is misleading. So conducting the extraction at 40 degrees C, which is typically represented, or worst case when you compare that to the long-term storage of the drug product, is in the case of large volume parenteral drug products where we're working at ultra trace levels, a good way to provide yourselves with an extractable data set that is a more accurate indicator of patient risk. And along the similar lines, immersing stoppers in a solvent system is not always representative of the interaction with the drug product when they're seated in a vial. For example, it doesn't establish how effective a barrier coating film applied to the stoppers on the drug product contacting surfaces is at minimizing leachables. So immersing the stoppers provides an interaction between the solvent system and the top of the stopper that would never be in contact with the drug product and often doesn't have a barrier coating. That approach is not relevant to how that vial stopper is seated in the vial during long-term storage. So again, may result in data that is misleading when it comes to risk. Again, solvent choice. Solvent that mimics the bulk properties of the drug product is more predictive of leachables than a hexane or IPA extract. Most LVP drug products are predominantly aqueous, so a hexane or IPA extract provides data that is incredibly misleading when looking to establish risk. So really the take home here is be clear on the intent of the extractable study and select conditions with that intent in mind. If the intent is to predict risk and predict the leachable profile, then consider a simulation study where you're not subjecting the container closure system to incredibly high temperatures, and you're replicating the interaction between that container closure system and the drug product. If the intent, on the other hand, is to chemically characterize a material like it is for medical devices, then okay, a more aggressive extraction study may well be appropriate and necessary. Okay, so along the same lines of sensitivity, really even the latest state-of-the-art analytical instrumentation, high-resolution mass spectrometers, coupled with mass spectral libraries and structural elucidation techniques, cannot really identify all compounds at such low levels. Some extractables, for example, rubber oligomers, are generated during the manufacture of the material or component. These aren't often commercially available, and so because they're not commercially available, there's the reference mass spectra of those aren't on the libraries that we routinely use to identify these compounds. And so given the complexity of the reaction mechanistics during the rubber curing process and rubber manufacturing process, it's incredibly difficult to predict how these compounds would have been formed and therefore what their structure is. So we have to accept that the lower we go, the greater the likelihood of unknowns are, and analytically, we are going to have to accept that in some situations it's not possible to identify them and derive a structure. So thinking about how we might manage that risk via acquiring other knowledge, and doing other studies is really quite important. I mean, along the same lines, there are structural elucidation tools that we use, such as preparative LC, where we essentially isolate the peak of interest, in an amount that's, um, then enough to conduct NMR on it to establish a structure. That's not an option for these compounds that are at such low levels. There's not enough to do NMR, and there's not enough to isolate it from the column. So the conventional tools that we would use to elucidate the structure are not amenable to such low level compounds. So really, for those extractables or leachables that cannot be identified, it's not possible to assess the patient risk using the analytical data. So we need to look towards other means to close that gap and understand what the risk is. And this is why it's important to work very closely with your preclinical colleagues or your toxicologists to understand what alternatives you may have to analytical data to further support the safety of your container closure system or stopper. So biocompatibility data provides you with more knowledge, working with the supply chain to understand the formulation of the material, um, gaining or acquiring some of the compliance statements and testing they've done provides further evidence that there are no chemicals that are present within the material that present a significant toxicological risk. So the take home here is that it's unrealistic to expect an analytical chemist to be able to identify every extractable or leachable compound at such low levels. So recognizing the role in vivo, in vitro biocompatibility data and other forms of supply compliance can play in supporting the safety of the packaging system is really quite critical when it comes to LVP systems, if the intent is to provide a robust package to demonstrate product safety. Okay, on to the final few slides now. Talking about leachable studies now, and really these should be some risk-based considerations that you should have when thinking about your leachable study. If the AET is very low, and therefore the sensitivity needs are high, we need to bear in mind that the drug product itself can impede sensitivity by interfering with the analysis. So something to think about when designing your extractable studies. Are you likely to be able to achieve the AET by analyzing the drug product? If you can't, then you need to focus your efforts on the extractable study design to try and discharge the risk in that way. There's a couple of options when it comes to leachable studies. Either you can use non-specific methodology like you would use for extractables, which are designed to be able to detect a wide range of compounds, or you can use highly optimized targeted methods that are typically more sensitive because they're focused on targeting one or two compounds. So those are the two options. But if the outcome of the toxicological risk assessment indicates there's negligible risk, really the benefits of a non-specific leachable study outweigh the risk of a highly accurate targeted method, because you can address the risk of leachables from multiple sources, from the manufacturing process, from the container closure system, perhaps from secondary componentry that you haven't subjected to an extractable study. And then when it comes to time points, batches, and storage conditions, again, if the outcome of the toxicological risk assessment indicates there's negligible risk, consider the benefits of including multiple batches, time points, and storage conditions. If you're using the same batch of stopper to manufacture 3 batches of drug product, then really there is no reason or significant reason to believe there's any variation. If you're using 3 batches of stopper to manufacture the 3 separate drug product batches, then there's a source of variation there that you may want to consider exploring if the risk warrants it. And really, resource investment, i.e. the extent of leachable testing, should be commensurate with the perceived toxicological risk. And doing a lot of work up front as part of the extractable study design provides you with that justification and understanding as to what the risk is. Okay, so here are some topics that I feel as an industry we need to start thinking about. Definition of sensitivity. So we've got to recognize that sensitivity of screening method applied to extractable studies or leachable studies is only an estimate when comparing against the AET. And it depends very much on the approach in which you've used to define what that sensitivity is. And I think it would be great if we could standardize the process for estimating the sensitivity of extractive methods. Could we agree as an industry the internal standards on which to base and define our sensitivity to really level the playing field? I think as an industry, we need to recognize that some LVP drug products either have no MDD or one that's so high that it's impossible to align with an SCT of 1.5 micrograms per day. So we need to start considering the practical benefits versus risk by capping the MDD at 2 liters, maybe in line with ICH Q3D. I don't see any reason why an organic leachable is any different to an elemental impurity. So capping the MDD at 2 liters appears to make sense to me. And I think as an industry, we need to think about that to overcome some of the significant challenges we face. And then unidentifiable extractables and leachables. So we need to recognize that an analytical chemist doesn't have a magic instrument that can detect and identify all potential extractable leachable compounds at the AET for some high daily dosing drug products. And be receptive to discussing, agreeing solutions to this challenge to ensure that the level of patient risk is understood to the best of our knowledge with the tools that are available, and balanced with the clinical benefit of the product. And that concludes my talk. Thanks to everyone for your attention, and I'll take any questions. Thank you.